So again, two parts for photosynthesis. We're first going to talk about the light-dependent reactions. Now, before we get into that, I think we need to talk a little bit about the chloroplast itself. Again, the chloroplast is an organelle within plant, algae, and some bacteria tissues where they can actually store energy emitted by light. And we'll talk about how that's done, but let's talk about the inside the chloroplast itself. There are these little green discs inside the chloroplast called the thylakoids, these cylinders or these discs in here, the green disc structure. The lumen is the space inside the thylakoid. So you see some cross sections of some thylakoids here. The lumen is this yellow space inside. Now there is the stroma, which is this liquid material inside the chloroplast that surrounds the thylakoids. Now, not the stoma or the stomata, that is the outside, that's on the underside of the leaf that controls what goes in and comes out of in terms of gases and air and water, has some control on what happens with the leaves. That's the stomata. This is the stroma, which is inside the chloroplast. The granum or granum, that is where you have a stack of thylakoids. Right? We'll talk a little bit about why that's important, because why that's important, the light-dependent reactions occur inside one of these thylakoids. It occurs in the lumen within the thylakoid and also kind of on the membrane of the thylakoid. But a lot of what's going on is going to occur within the thylakoid, within the lumen space. Okay, so really what's happening is, I think I mentioned earlier, it's kind of the reverse of metabolism of glucose. It really kind of is. There are a lot of components just kind of running in a different direction. So an electron transport chain gets set up within one of these thylakoids. And what happens is we basically set up this hydrogen gradient inside the lumen, inside the space within one of these thylakoids. You've got these different proteins and different structures that are going to move high energy electrons throughout the structure and move hydrogens back and forth. And we've got our friend down here, the hydrogen turbine ATP synthase that will generate ATP. So let's look at that a little bit. I think I've got, yeah, so there's two complexes here that are absorbing the energy from the light. There's photosystem 1, photosystem 1 is over here, and photosystem 2 is over here. We call them 1 and 2 because, if memory serves, that's the order in which they were discovered. So photosystem 2, in both cases, these absorb light energy into chlorophyll. Okay? So energy from light radiation particles add energy to chlorophyll in these photosystems. Okay? And then this energy gets accumulated and passed between the chlorophyll molecules back and forth until enough energy is stored. So enough energy from the light gets absorbed into the chlorophyll, gets stored, spread out, and eventually we hit a point where we can actually excite some electrons. And what that means is we can actually strip off some electrons off of some water and allow the hydrogen and the oxygen to separate. So now I'm increasing the hydrogen ions inside here. Okay. And then we can set up this electron transport chain. So now I've pulled electrons out of that water, stuck them into this photosystem 2, and then I can pass those electrons to these different protein structures, which I'm not as worried about the individual names of the protein structures. I do want you to understand that they're pumping hydrogen from the stroma, the space between the thylakoids, into the thylakoid. They're pumping hydrogen ions into the lumen within the thylakoid. All right, so this is one kind of simplified model of a thylakoid we're seeing here. And again, light striking these two photosystems and allowing electrons to be passed through these proteins. And while it's happening, hydrogen is being pumped into the thylakoid, which is what we have here. So we're going to pick up that on the next video.